Um, so we're on our homepage, rockefelleruniversity.github.io. And it's here that you find links to our videos and, of course, all the training we are running in our sessions. We have so far introduced ourselves to the basic of working in R. So we have our introduction to R. We've done our plotting in R, which is mostly ggplot. And I showed you at the end a little bit about how you can mix some of the stuff we learned in reproducible R, effectively build an RMD and R Markdown, read in a table and make a ggplot interactive. We did that at the end of last session in the exercise. So now we're going to start to look at high throughput sequencing data. Okay, it's the main part of what we're going to cover. But before we launch into actually analyzing these in R, today's session, I don't think we have any programming. We're going to introduce ourselves to the type of data, where we can get this data to, and if we're producing it, where we're eventually going to submit it to. And that is in this genomics data slides. It shouldn't take too long. We'll do this first session, genomics data. I introduce you to some databases, and then in the afternoon after this next session, we will cover IGV. So how can we graphical user interface to start to investigate your data in a linear genome. So what I like about these two today is that actually some of you may never do any programming, you may have come here just to learn a bit. But from today, you should understand all the formats you're going to deal with, really what they contain, and then this afternoon, how you can just go and look at these in 10 minutes worth of messing around, get it into a linear genome, and start to investigate your data or other people's data, which is really half the fun. So we're going to start with genomic data formats. I'm going to try and go slow. So reference genomes and genomic file formats. So as an overview, I'm going to start by introducing us to the reference genomes. What are reference genomes? How do they actually look? If we download one and put it on our computer, what is actually contained within it? And then we'll start with Fasta and fastq. So this is probably the first two data types you'll come across if you're doing high throughput sequencing. You will need a Fasta to start your alignment, to start your analysis. And somewhere like Genomics will deliver to you fastq, your unaligned raw sequences off the machine. We're going to see how we can turn fastq into SAM and BAM. So these are often just the next step from unaligned sequences. We can use Fasta, the reference genome, as a Fasta format in combination with your unaligned sequences to produce SAM and BAM. And then we'll go through some of the more summarized data formats. BED, it's a very simple format that can contain a lot of information. GTF, GFF, this is how we handle not just genome information, gene annotation, but really complex genome annotation. Wiggle files, beg graphs, and bigwigs. These are very easy to review in a linear genome browser. They tell you scores at a particular interval. I have a lot of RNA-seq signal at this gene locus. I have a lot of openness, open chromatin at this particular location. Beg graphs, wiggle files will help you with that. And if anyone here is doing genomic variations, I think a lot of people do, we have BCF and MAF format. If you want to ask questions about BCF and MAF, I know enough. In my team, we have some real experts in variant annotation and analysis. And then finally, for those of you, and I imagine if you're doing any high throughput sequencing, you're going to be doing single cell. These two formats are the new single cell formats and certainly starting to become the standards. So we're just going to show you really what's contained within them. So, obviously the human genome is pretty complete, but it isn't complete. And if we looked, I don't know if you saw this announcement, but somewhere we had the Jarvis laboratory, they published something reasonably recent with the Human Pangenome Reference Consortium. And we're still trying to build in the last parts of these genomes, and that includes end-to-end sequence, going through the really hard can map regions like telomeres or centromeres, as well as accounting for variation in the human genome. It's certainly not done. In fact, most model organisms references, despite being the focus of attention, they're not complete in their reference. Okay? Certainly getting better all the time, but they are not complete, and they are regularly updated. So reference genomes consist of a mixture. So these things like the human genome, if we were to try and download it, it's a mixture of known chromosomes, things like you would recognize chromosome 1, 2, 3, 4, 5, and what we can call unplaced contigs. We know this bit of sequence belongs together, but we don't know whereabouts in the genome it actually goes. Okay? And that might be because it's too variable. It could go to multiple places, or it could just be we haven't managed to assemble the genome fully enough yet that we can place this contig in there. So we know the sequence exists somewhere in the genome, just don't know how we can place it in a karyotype diagram where you have all the chromosomes. We have two types of revisions for these genomes. So we have major and minor revisions. So major revisions, it's going to say here, it's going to result in a change in coordinates. It's more serious than that. Major revisions, you're pretty much going to start again with your analysis. You're going to take your sequences, you're going to realign them to this big change in the genome, these major version, and then start again. And that's because major genomes, major revisions, they will often find all of a sudden this unplaced contig. Now with better sequencing, longer reads, I can now place this confidently in the middle of chromosome 2. Everything in chromosome 2 shifts along after it. Coordinates change, and we have to do some realignment. There are tools to do what is called liftover. I don't think we cover that, but I'm happy to show you. And that allows you to map from one major revision to another, but typically we're going to start again. Okay, you need some type of conversion between these revisions, either using this liftover tool, or most often, I recommend you just realign your data to the new version. I think the new version of the human genome is still GRCH 38, sorry. And alongside these major revisions, we'll also get minor revisions, and quite a lot of them. Minor revisions do not change the chromosome coordinates. We don't necessarily need to start again, but they may add or change the annotation. So this gene is no longer called this gene, or this exon doesn't really belong to this gene, so we're going to remove it from the annotation. This gene's actually shorter than we thought. These will all be included in patches. So we may have GRCH 38. We may be on the 6th or 7th patch. So yeah, when you download a genome, you'll typically download something like this. You'll know its major version, and you'll know its minor version. This Genome Reference Consortium, I have the page open. Yeah, they're obviously a big collection of institutes that all work together to try and curate and maintain the reference genomes for the model organisms. And that includes things like this pan-genome project. There's like 10 major labs involved in that. I say 3 model organisms. I think it's expanded, to be fair, to include rat now and chicken. So if you are in the Jarvis lab, you've got chicken as a main model organism. And we have GRCH 38, GRCM 38, this is the mouse. And then we'll have things like the zebrafish. I think chicken and rat are now major genomes too. But if you're not working on the major genomes, you still find that often other laboratories are. Things like Drosophila, they can be maintained by the Berkeley Drosophila Genome Project. I think mosquito, we have Vectorbase, but you may know Leslie Vossel's lab. Previously done a lot of the annotation and the genome builds, especially for neurotranscriptome. And we work now with the Crone lab, a guy called Kip Lacey. And they built the I always get this wrong, colonal radar amp, despite working on this for months. I always get this wrong. And again, this is a Rockefeller University. They built the genome, they submitted it to NCBI. It's not maintained by a consortium. Without digging at the genome, you know, it's very different, the level of annotation compared to human mouse. But there are model genomes available, or at least genome references available for lots of species now. It's a lot cheaper to sequence a whole genome. And if anyone's interested, Olivier is giving a talk. He runs our reference genome, what are they called now? Reference Genome Laboratory, I think. Reference Genome Resource Center. He runs that, and he's giving a talk on Wednesday about how he builds genomes. So if you want to build your own genome for your own model species, talk to him. But lots are available already if we search around. And the reason we want a reference genome is because previously, when I was doing my PhD, we were relying a lot on express sequence tags. We could take the whole RNA, we could make cDNA, and we could sequence that perhaps, and that was really about the depth we had. So we could do things like tell a gene was better expressed than another gene, but what we didn't have is gene A and gene B are next to each other. If gene A, B are regulated by the same element, the same, say, enhancer or enhancer cluster, knowing that these two are close together, once we know the whole reference genome, is brilliant. Okay. Could be that they're both inside a TAD, some topologically associated domain, some type of transcriptional unit. We know this now by having the full linear context of the genome. When I say linear context, beginning of chromosome 1 to the end of chromosome X. We know where it sits in the genome. A really important thing for us is also by having a reference genome, which can cost a fair bit to sequence and certainly relies on a lot of reads. Now that someone else has built that for us, we can just do much shallower sequencing. Right? We don't need to sequence the whole genome. We can just sequence the RNA, and we can use the fact that we know the full genome to then place our little bit of RNA sequence, see where it maps, see where it matches on the main gene. So we can do a few millions of reads, use the reference genome, and start to get interesting observations, hopefully. So the basis of high throughput sequencing really is something like this. We take our DNA, so it could be your full DNA. We produce cDNA, so we make a copy of every transcript by reverse transcriptase or whatever. And once we have something stable like DNA, we can fragment our long pieces of DNA into short pieces, fragments, somewhere between 100 and 1,000 base pairs often. And then using the Illumina sequencer, and Connie will come in and give us a short talk about the Illumina sequencer. Connie's our head of genomics. We can often sequence just the ends of these fragments we've created from our cDNA or DNA. We sequence just the ends, and sometimes we can sequence these knowing they're paired. So I know that this sequence here and this sequence here come from the same fragment. But just by having portions of these sequenced, I can then use a reference genome and say, this bit of sequence here matches here. This bit maybe matches here, and then I can be confident that we have a fragment in my original data originating from that part of the genome. Okay, so that's why we really want a reference genome, is to allow us to take our aligned, our newly sequenced reads, and then place them as unaligned sequence now onto our reference genome. The reference genome is actually kind of simple once you download it. It's just a text file, often compressed because it's big, but not that big. I think the whole human genome is probably less than 10 gigabytes for sure. And a reference genome is really just a collection of contigs. I'm going to say contigs a lot. Then I can talk about chromosomes and unplaced contigs too. And a contig is just a stretch of DNA sequence encoded as an A, G, C, T, or N. So we still have unknown bases in the human genome. Not sure exactly which base is at this position. It could be a T and A. We know we have a stretch of sequence there, we just don't know what it was. Typically, and I think almost always, a reference genome will come in this format. So if you're opening it up in a browser, in text edit or Word, it will look like this. And it's really made up of a few components. We have a header line, which is this, greater than symbol, and that's sort of the beginning of a config, right? So this says, gives me some information on here, I'm not quite sure, but Homo sapiens, chromosome 1, and it tells me the actual name of the reference assembly. And that means I will have this line, this information, and then I'll have the whole of chromosome 1 as a sequence, all the way till completion, and then I will have another line like this, it's greater than, and it will say whatever, Homo sapiens, chromosome 2, and then it will have chromosome 2, chromosome 3, chromosome 4, and then we'll have the unplaced contents. So that's what our Fasta looks like, and that's what we're going to need to provide to our aligners in order to align the data. And if we want to view something in IGB, a custom genome, we're going to need to provide this too. Brought back. Well, your charger didn't melt. That's a nice change. In high throughput sequencing, we're typically going to get two formats provided to us. Usually, the formats you're going to get is the unaligned sequence files generated almost directly from the Illumina sequencing machines. You may, depending on how friendly your bioinformatics core is or your genomics, they also may produce a SAM file for you, which is the aligned sequences. So it's the same information as you would contain in the fastq, except additionally it has this bit of sequence belongs on this chromosome with this mapping accuracy, etc. So have a look at fastq. So fastq is Fasta with qualities. There were a few different formats, but now this is basically the only format you'll ever see from short read sequencing. We again have a header line. So for Fasta, we had this greater than. For fastq, we have the header line, and it starts with a at. So this is a header line. This is a header line. This is a header line. And every single record corresponding to a sequence we have generated is 4 lines long. So we have 4, 4, and then 4 here. Each got their own header line. We then have the sequence information. So this is the actual sequence as the Illumina machine has called it. Just there in that second line. We have this plus, which used to contain some more information, but I think just got ditched. So it used to be plus would contain basically the header line with a little bit more information, not anymore. Now it's just a plus. And then we have quality scores encoded as ASCII. I'm going to come back to that. But effectively, for every sequence, as you can see here, we have paired with it another ASCII character. So the first one is an at here, and then for the last ones here, we have hashes. Okay? I want to come back to this in more detail, but these quality here, effectively telling you how likely you are to have called the correct base. And the reason they're going to encode it in ASCII is because that saves a lot of space. We don't want to put 10 zeros, 0.1001 chance of this being wrong. We can encode it as an ASCII score, and I'm going to show you this in a bit. So the first useful line, and it's going to save your project occasionally, is the header line, right? Because this tells you whether you've messed up anything or got the wrong fastq. It tells you the machine you were sequencing on. And actually, this last bit of it here is also unique to your run. So this machine name here tells you I sequenced on a particular machine in the world. Okay, so you know this fastq saying came from our genomics center by matching up this header. You can actually tell how it was sequenced to. This is HiSeq HS, and there's code here. So I think if it says an X, it's a NovaSeq, and there's like a code you can work out from here. We don't need to go into that much detail. We could just say, this is our machine. We then have the flow cell. So it's, Illumina updates a lot, but I used to have 8 flow cells in some of their machines. So you could be in any of these 8, and it would sequence all 8, and we would get results from the different 8. Now I think it's just 2. I don't think they actually run sequences, but this tells you that. And then this, well, this tells you whether it was read 1 or 2 in a pair. So am I at this end? This would be read 1. This would be read 2. But they came from the same fragment. So that's important when we're mapping. We know that they were originally from the same fragment. And then, I don't really explain it here, but actually this 1001 and 1595, 156011, that effectively tells you where in the flow cell this particular sequence came from. Okay, so a flow cell is a 2D surface. It's imaged in order to give us sequence information. This just tells you where it was. The real importance of knowing where this read was generated really is if I start to see lots of low qualities and all the reads are in the same place on my flow cell, that tells me I have a positional effect. Okay, so my top of my flow cell was ruined. I can actually extract, the sum qualities. We'll do this later. I can sum up the qualities and I can find the position and say, this area of my flow cell was ruined. I throw away all the reads from there. We then have the quality line. I'm not sure how much detail I go here. But let me bring out good old Wikipedia for this. There we go. So Wikipedia has a really nice script. Okay. So what we do here is for every one of these bases, we effectively have a probability of this base not being an A. Okay. So we want to be able to capture that information and store it efficiently. What Illumina is going to do is it builds like a range, bins the probability into about 40. Okay, then it assigns each of these bins an ASCII character. So hash may be the lowest level of quality. B may be one of the highest levels of quality. By doing that, we can save ourselves a lot of space. We don't need that level of resolution really in our quality. We don't need to know the difference between 0.1002, 0.1021, right? So we can bin this into a nice bit of ASCII space. And then later, whenever we pass this fastq to our aligner, although it's hard for us maybe to by hand translate E into a probability, the computer's going to have no problem. So it's going to do it instantly, effectively, and then be able to say, well, a read with a very low quality, I don't trust. Maybe a read with a really great quality, I do trust. We can also use this quality, and you can often notice, here's kind of an example. Clearly, hash is the lower quality because we have lots of things ending with hash. Often in Illumina sequencing, the reason you start to drop off in quality is just reagents start to run out. The machine has a certain amount of reagents to try and push this. So you'll often see quality drop off over time. And again, we can use the quality scores to say, oh, when did it start to get bad? And we can start to do clipping based on these quality scores. I'll throw away the last 10 bases. We're going to see that when we start doing a bit of programming. There are tools which will just, you don't have to do any programming, pass the fastq. It will build like maps based on this quality score. I did want to show you this. If you really want to know more information about these quality scores, for some reason, Wikipedia has an absolutely fantastic map of kind of the range of quality scores. So if we looked here, I said hash was probably bad. Here's hash at the very bottom. Exclamation mark's probably worse. Hash is pretty bad. And then this is the maximum range for this, which is Sanger. Selects is there. And so the highest score for this was an I, equivalent to the 40th bit. So it's not high resolution, but it gives enough of an idea. This is poor, this is good. Okay. That's all we need to know about fastq. So the next format we're going to talk about is aligned sequences. And I can tell you, back when I started doing bioinformatics, there was no standard format, and it was a nightmare. Everything had a different format, and we would end up converting to really basically text-like formats. SAM format was done by Heng Li, who actually is super famous. I just noticed he was on this paper as well. So Heng Li's in Sanger in Cambridge in the UK. So he writes lots of essential tools, and one of his real contributions was SAM files, sequence alignment map, and the specification which went in them. Okay, this has now become the standard format for sequencing data. Although we can call it SAM format, you will likely see a file called BAM. Right? Sequence alignment map, that is binary alignment. It's the same information, it's just compressed. Right? So it's compressed so a computer can read it, we can't. If we turn it into something readable, it will be SAM format. Majority of software and browsers recognize SAM format. In fact, it's just the standard now, so you're going to need to create it. Okay. So despite the fact that there is a binary version of this, it contains the same information. We're going to talk through the SAM, because this is what we're actually read if we wrote it to a text file. So SAM potentially contains a lot of information. We talk through some of the most useful things. The first thing is at the top of every SAM file. So first, however many lines, will be what we call the SAM header. And this really tells you what is coming up in the SAM file. So we have a few important tags. We have the HD tag, and this tells you the version number of the SAM format. SAM format has updated over time, but typically it's 1.4. We also have here, I'm going to come to this in more detail, SO, which is sort order. So when we have a SAM file containing all our reads, we can organize that SAM file so the reads are arranged by their position in the chromosome. That would be called coordinate sorting. We can also organize them by the read header, right? And that's query name sorting. Okay, but we don't need to actually check the SAM file, and it can be computationally expensive to check the SAM file. Is it all sorted? We can just read the first line, and it tells us this file is sorted. If it says unsorted, we'll bother sorting it. Below that, we then have the sequence information. So we have the actual sequence name. So we can have chromosome 10 down to chromosome Y. And then we have the length of that particular chromosome. So another really useful thing, if someone gives you a SAM file and you're not sure which version of the genome they've aligned it to, knowing the length of chromosome 10 in the latest human genome versus the one 2 years ago, you can use this header to actually check. Oh, wait, they've aligned it to HT19. I need to update it. Okay. Another useful thing about this is the actual order in the SAM file. So if it's sorted, we know it's sorted within a chromosome, the position. It goes from, say, the start of chromosome 1 to chromosome, end of chromosome 1. But then the chromosomes in this file will be ordered 10, 11, 12, 13, 14, 17, 18, 19. So any reads on chromosome 10 would come first, and then any reads on chromosome 11. So without having to read the file, we already know a little bit about what's coming up. The actual SAM file itself contains both the fastq information and the alignment information. So everything we had in the fastq is still here, but now we also have information on the alignment and the position. So just to show us, we have our header here. This is a different read than we looked at earlier on, but machine information, flow cell, position in the flow cell. That's all there. We have the actual sequence itself, still stored in the SAM file. And then we have the encoded sequence quality here. So already, what's something worth pointing out is that a SAM file you can use for new alignments. Right? It has more information, it has all the original information in the fastq. Right? So it has the sequence information, it has the encoded sequence quality, and it has the header. I can recreate my fastq from the information in here. On top of that, it also has some really useful information on the chromosome that's aligned to, the position it aligned to, the 5' of the read. So the actual start of the read, where that aligned to. And then we have this here, which is our cigar string. So this is our first example of run length encoding. Rather than writing out 100 matches in a row, this matches 100 times, it just writes out 100 M. And that's already saving some space. Here we have something a little bit more complex, and if I move this down here. So here we have 28 matches, 1 deletion versus the reference. Right, so my file's perhaps missing a base which was in the reference. And then we have 72 matches again. Okay, so that's again encoding quite a complex alignment, and only a few lines here. Okay, we probably don't need to read this ever. We're not going to go through these files one read at a time, but the computer will go and read this SAM file, and that's how we build visualization of BAM files for things like IGV. It's also how we find splice junctions, because there we'll have map, and then we'll have a gap, and we'll have more. On top of that, we also have more useful things encoded in here. We have the position of the paired read. So if I had pairs, I knew read 1 was on the same fragment as read 2. When I look at read 1 in my BAM file, so this is the position of read 1, it will tell me where it's pair mapped and the distance to the pair. So that's going to be very useful for when we do things like attack seek, and we care how big our fragment sizes are. We're going to use it there. We also have another way of encoding information about the alignment stored within this bit flag. So I'm just going to go to this little site here. Where'd that go? So by using that number, let's try and type this in here, 147. Oops, 147. You know, obviously we wouldn't do this one read at a time, the computer would do this. But then this little number here, encoded in it, is telling me the read's paired, it's telling me the read mapped in a proper pair, it's telling me the read mapped to the reverse strand, and it's telling me it was the second read in a pair, so it's from the other end of the read. So we can get that just in 3 numbers, right? It tells us all that information. Okay. So that's all the information we have in a SAM file. Remember, we're going to encode this as a binary alignment map later. Yes. So if you look here, this 100, well, we have the sequence here for a start, so we can just look at that. But the 100M, right, you can actually, that is based on the read. So we know it's 100 match there. And if the one which is a bit more complex, here, 28 matches, 1 deletion, so the reference had something, and then we have 72 matches. So 72 plus 28 will give us the total. Because actually from here, we can both find the read length, but also the length of the position it covered on the genome. Makes it really cool. Good question. Can I go a bit faster, maybe? When I first started doing bioinformatics, I never saw a fastq or a SAM file. I just saw bed files. So really with that SAM file and with those fastq, we're trying to get it placed on the genome, but we may want to summarize it just that a little bit more. I might want to say, I just want to know the number of reads which have aligned to, say, a gene, the full gene position. That's going to be much simpler. It's going to tell me the start of the gene, the end of the gene, and then maybe give me a score. How many reads do I have at that position? That's the type of information we would store in these genomic kind of summarized features. So bed, which is the simplest, wiggle and bed graph, which is actually still very simple, and GFF, which is effectively a bed file, but allows you to arrange groups within your information. So this is the first format of bed files. This is bed3 format. And it's very simple, chromosome start end. So I have a feature which starts here at chromosome 7, sorry, on chromosome 7, starts here, and I don't know what the feature is. Right? I don't know if it has any score associated to it, but in simplicity, I know I have a feature there. So that's bed 3. Bed 6 is probably a little bit more popular because it has that much more information. So bed 6 is basically bed 3, chromosome start and end of the position. But we have a name, which is useful. So that could be your peak name, your gene name. We have a score, and this can be any type of score. It doesn't need to be a whole number. And then we have a strand. So this allows us now to have a gene on one strand and not another strand. So this bed 6, they are just tab separated files with these lines. You can create them in Excel, save them, and then import them into other programs. If you just have strandless features, and like if we're going to do ChIP-seq later on, we will do ChIP-seq later on. ChIP-seq is strandless most of the time. We don't know if it was on the positive or negative strand. Often people will put a dot if it's strandless. Another equivalent is actually a star. If you put a star, it just means we're not sure of the strand. ENCODE, so if hopefully some of you know what ENCODE is, big consortium of really genomics data across multiple species, and they provide lots of different BAM files and bigwigs and bed files. They come up with their own format, which is really an extension of bed 6. And it just allows them as narrow peak and broad peak. These are used typically for ChIP-seq. It just allows them to additionally contain p-values and q-values. Right, so they can tell you the actual significance of a particular peak. The really nice thing about narrow peak and broad peak, you'll see later on, it's integrated well with IGV. So if we load a broad peak into IGV, it knows I will make the color darker depending on significance, lighter if not. It doesn't do that with bed 6. So that's actually kind of nice about those formats. One of the formats I think is most useful is the idea of these wiggle and bed graph formats. I still have no idea why it's called wiggle, other than it makes a kind of wiggly line across the genome, I guess. Bed graph makes a lot more sense. Okay, so these two file formats, again, the underlying text will be wiggle or bed graph, but we're going to compress these to something which is computer readable. So wiggle format definitely fell out of style, but we're going to see big wiggle later on, which is still the most popular. And wiggle format is effectively all it has is a header line for every chromosome. So here it's saying it's variable step. I'm not sure how true that is. The chromosome's chromosome 21, and it's going to be span. We're going to do windows of 5. Okay, and you can see then it writes out the first 50, and then it writes out 5 base pairs later, the coordinate 40. So it's saying from this position till this next position, we have a score of 50. From here to here, we have 40. From here to here, we have 60. From here to here, we have 20. All the way through the genome in steps of 5. So knowing we're at 941, 9,411, 1,306, it's going to be a big file, right, stepping it through. Bed graph makes a lot more sense if you have sparse data, because you can imagine no matter how deep you sequence, you're going to get lots of zeros. I just don't have reads in that particular region. It's going to be a 0. Bed graph accounts for this much more sensibly by having true variable step. And it's basically a bed 3. with one additional column, which is the score. And this can be any score, it can be decimal, integer, whatever. But it's also a variable step. So in this last line here, we go from 10,051 to the end of the chromosome, say. There's no signal, but I can contain that in one line. Here, I would contain that in 100,000 lines or something stupid. So bed graph is a much more sensible text format for this. And again, you can create these in Excel, import them into your IG. I'm going to show you the binary versions of all of these here. The next big format is GFF. And this is how, if you're doing any RNA-seq, if you're doing a TAC-seq and you want to have your annotation, it is going to come in GFF2 or GFF3. GFF2 being GTF format. They look very similar. And basically it follows this chromosome. Do I have the actual breakdown of this? So yeah, this is used to store genomic information annotation. It's absolutely the format which has won the storing annotation. And it stores position, the feature type, and importantly, the meta feature type. So I know this exon belongs to this gene. That's really the most important thing about this. So to show you, we have chromosome here, start, end, and then strand. Okay. So everything we have from our bed 6 is there. We also have score and name, I think, here, but it's just not, they're dots because I don't have anything in this position. Ultimately, we have source. So if this was generated by BLAST, that's good. Often this will say ensemble or UCSC consortium. Occasionally it will say the lab, which has added this particular gene. The feature type, this is important. In GTS, we'll come to this. UTRs, exons, CDSs, that's what we might have in this feature type here. Gene transcript, we can have that in here. And then we have, yeah, the score column here. Here I have no scores assigned. The thing which is most useful for us using this is that it has this idea of relating a feature to a meta feature. So in this case, this exon is a part of this gene. It could also be that this exon is a part of this transcript, and that's also a part of that gene. I can have any number of meta relationships here, but importantly right now, I know that all these exons correspond to the same gene. That means that when I go and use this in RNA-seq counting, I know to add up all the scores across these exons and place them into one value per gene. Simple as that. It also means when we load this into IGV, it's going to know to connect those exons together. The last couple of formats I'm going to look at is VCF math, and then I'm going to cover HDF5. So does anyone here do SNPs? Is anyone working on genome annotation? Yeah, so VCF is a super important format. VCF is typically just a text file, mostly likely stored in a compressed manner, and we'll talk about that, I think, in a few slides. A VCF contains meta information lines, a header line, and then importantly, the data lines. VCF actually allows you to also contain the information from genotyping experiments. So information on every single sample can be contained in a VCF. You can go in there, look up your mutation of interest, and then see which samples are assigned that particular change. What I really like about VCF format is it explains itself, which is always good. So if we look at a VCF, this is it in text format. We can see we have fixed information here, genotype information here, so samples are along here. Okay. And then we have this metadata information here. This is what tells us what is coming up in the rest of the file. So computers will read this bit, and then they will translate what's happening in the rest of the file based on these descriptions. Also notice we have our comment characters here. Right, we have one hash here, right? So if we did the read delim, it would ignore all these lines if we just read this in. And that's deliberate. They're following a convention here. And they're following an interesting convention where they will have two hashes for lines you definitely want to ignore. But if you want to get the header line, that's got a hash as well, but just one hash. You can actually distinguish between the two. I might want to not read in the header line. I know what I have. Or I do want to read in the header line, but I don't want to read in this. You can kind of control that with different comment chart settings. So just, I don't think I have any more on this slide. So just to talk it through, we have chromosome position. We have an ID, so known SNPs. There's great databases for known SNPs. And here we have an RS. I think that's a dbGaP SNP identifier. You could Google that. If we went to NCBI, there's a database explaining that SNP, all the diseases associated to it, where it was found. We have the reference allele of that position and then the alternative. Okay. We have quality, we have whether there's passed filter. Often if you're running GATK, these machines which generate VCF from a BAM file by comparing the reference to the sequences we have in the BAM file. They have various filters which really allow us to decide, do we trust this? General information here, but if I didn't know what this meant, I could just go up to my metadata, AA, that's the ancestral allele, okay, AC, total number of alternative alleles. Okay, so this is actually free text here. They can have pretty much whatever they want to describe from the metadata. What's really important is often they will have DP, which is the depth. And you can see the depth in every sample. If you go along here, we can see format GTDP. This is genotype in that sample, and then the depth in that sample. So from just scanning along, if we were reading it by hand, we can say this wasn't a gene, this wasn't a mutation in this sample, wasn't in this sample, wasn't in this sample. But here, this line, we could say one of the alleles was different from the reference. It went from a T to a C, and the total depth in this sample was 25 reads. So there's a lot of information in a VCF. And really, we can actually translate it straight from the metadata to see what's happening in the rest of the file. I'm going to show you the VCF binary format also. Another format I don't really use much, but JD thinks is great, a guy on my team, is mutation annotation format. So that's MAF format. It's effectively further than VCF. It's called aggregated mutation information. And it contains not just the SNP, but a little bit of information about that variant in particular. So it might add some variant classification saying it hits a splice site, saying it causes a mutation, a missense mutation, or in fact, it has a silent effect. It doesn't actually affect the protein coding at all. Okay, so MAF is effectively an extension of VCF, where it's more gene-centric. It contains not only information on the gene the mutation may be in, but the potential impact of that change. Okay, so I think the last few slides here are just talking about really compressed versions of this. And it's just to say, all of these formats we've looked at, there is a compressed version. I like to be able to understand what's in the file. You will at some point probably need to, if you're going to do some serious programming, you want to need to work through these things line at a time. But for all these formats I showed you, we have a binary equivalent. For SAM, we have a BAM file. I'm going to show you how you can turn a SAM into a BAM file as well, and a BAM back into a SAM. A BAM file is a binary alignment map. It will come with a .bam extension. To really make use of a BAM file, you need to index it. Sorry. You need to index that file. That means running another program, and that produces a binary alignment index. Right, with those two together, that actually means we have random access to the SAM file. I can say, give me the reads from chromosome 10, this position, and it can go look in this BAI file. It knows roughly where to start looking by creating that index, like a real index for a book, and it will go just pull out the reads from that region. And it is millions of times faster than going through the SAM from line 1 until I get to that particular read, that region. So once we've made a BAM file from our SAM file, random access is a lot quicker. Wiggle and beg graphs, I showed you them. Big wig is the format you will see. It's often .bw, .bigwig. Beg graph, big bed is a very popular format. That was created by UCSC. So wiggle was UCSC. So bigwig is UCSC. Bed to big bed, that's another UCSC format. But actually, probably the better format is called Tabix. And Tabix is basically allows you to create an index, a TBI, a Tabix index file from anything which has chromosome start or end. So if you have chromosome start end, we can Tabix index it, and that means we can randomly access. If I want to know mutations over this particular area, I could then use the Tabix file to quickly get it. If I want to get the genes over a particular area, if it's Tabix indexed, it's super fast. These are also not just useful for our programming. When we want to use IGV, IGV doesn't want to load the whole file in memory. They just want to load the window we're looking at. So we'll see that very soon. Okay, so maybe this will be the last thing. Okay. Okay. So as we have started creating bigger and bigger experiments, the results of many of these experimental assays has actually become a little bit unwieldy. And in particular, we're talking single cell and single cell RNA-seq, attack-seq, or nuclear RNA-seq. The results of many experimental assays is actually usually just a combination of tables. And it may be the table of measurements, say gene by sample. Okay, so I may want to know in single cell, for every cell across my columns, what is the expression of a gene? I may need some metadata to say this cell came from this experiment, this cell is in this cluster, this cell has this particular phenotype. I may want gene-to-gene information table, how genes relate to each other. I may need to actually transform my data and still save that information. And that could be as simple as log2 transforming my data. If I want to arrange combinations of tables together, I can either do that with a folder and have 3 Excel files or 4 Excel files. But when we're talking, as anyone probably has experience here, when you start trying to open millions of rows in Excel, Excel dies. We don't need to, right? So just an example of this would be our samples here, genes here, sample names, what group they're in, maybe the RIN score, and then gene ID, and then symbol. This is information on the genes themselves. With these bigger data sets, we want a format which is easier to share. And as much as I'm going to try and convince you all to work in R, some of you will go work in Python, you want a data set which you can transfer from one language to the other. Maybe I got better bits in R, better tools for me there, but eventually I want to do this display in Python. If I could have a format I could save to and it's readable in both, that's clearly an advantage. And there's also an advantage for collaborators. There are some collaborators who will say, I just want to do this part of your analysis. And you go, thank you. What format do you need? This special Python related format? Then you have to learn that. We want a cross-platform type of object. We want it to be able to handle and connect different data types. Really importantly, it needs to be fast, memory efficient retrieval of the data. We don't want to, every time we look at this file, my laptop explodes and it takes 20 minutes just to pull out one value. So we need some of the advantages we're getting of BAM files. And actually this group, the hierarchical data format, I'm not really sure how these groups come about. They basically set about building a new type of data format for high throughput computing, not just about bioinformatics, not just about single cell, but how can we store hierarchical data, produce a type of format and the tools which go with it, which importantly describes itself, right? So I can have a format which somewhere in there, it describes what's in that object. It is cross-platform, it is memory efficient and fast to pull information from, and it works well with large data sets. And the HDF5 group have been working for some time to produce this type of object. For us, this object has now become incredibly useful for single cell, single nuclei, attack-seq and RNA-seq. Okay? Because there we have a very large matrix. And I'm not sure if has anyone done single cell here? So you're starting to understand the sizes of some of these tables. And if you try and write out that matrix just to a text file, it's gigabytes often, right? It's very, very, very big. So it's impractical just to operate like that. So Loom format is an extension of HDF5. So it's effectively HDF5 with some rules built around single cell. Okay? And it allows you to store the main matrix, so the counts, cell to gene, row attributes, such as my gene IDs and symbols and information there, column attributes. This is typically sample names and sample attributes, graphs, and if anyone's done single cells, things like UMaps and the connection between them, and any additional layers. Okay, so the Loom format, although I can't physically look at it in a text file, this is the format that we're going to start to learn to use for single cell. Okay. Again, it's implemented in multiple languages. I think the Loom came from Python, to be fair, and it got picked up by R. But it works really well. We have LoomR in Bioconductor, we'll show you, and you can just import in R, change the bits you need, and export back out to Python or C or Java, whatever else wants to use. Really importantly, it's fast and memory efficient. I can have really big Loom files on my computer. I can pull out just the information I need. I'm never taxing more than a few gigabytes of RAM. So that's the end of this first session. If you don't mind, I'm just going to go 10 minutes over to show you a little bit of the second session. I don't have any exercises for this first session. So if you don't mind, I'll just take up a bit of time. We'll do it. If you want some more help on these formats, we have UCSC, our formats here. So that's a good link. I think they talk you through all their major formats. What they have to say. IGV, we're going to use that later on. It's very useful. And then GFF actually came from Sanger. GFF2 is deprecated, but I think we were talking about GFF3. And you can tell you like the low-level description there. Maybe I need to link to GFF3. So I'm just going to quickly go into our next session. It's very so many of us actually won't be generating our own high throughput sequencing data. Right. As I say here, it's the third option, by sequencing some of your own material. But often you can just get hold of high throughput sequencing data, and I really recommend this in home studies, from public data, public repositories, people who are your collaborators or competitors. You can go and look at their data and see how it fits in the context of your question. Okay. So high throughput sequencing data, by its nature of producing it and trying to publish it with it, it needs to be submitted to a public repository. Most journals will force you to do this. They won't accept it until you have that ID. So we're going to talk a little bit about some of these public repositories for high throughput sequencing. So some of the major ones is GEO, which is the Gene Expression Omnibus. Maybe you've seen this previously. We have SRA, which is the kind of sub-GEO, but it's built just for short reads, so just for archiving fastq data. And then we have the European equivalent, ENA, which obviously I'm familiar with, but also has some features which make it kind of appealing versus some of these other ones. So GEO holds different types of biological data. It is not built for sequencing data. And that's the first thing which is slightly problematic about it. It was originally built for EST arrays, cDNA arrays, gene expression data. That was microarrays, that was Affymetrix. So it's had to adapt to GEO, and it's done a good job. But it means that it doesn't really capture in a uniform way information about high throughput sequencing. So it captures the metadata, the processed files, and raw data by way of SRA, because they're linked. What it does very well is actually capture kind of free text descriptions. So I'm just going to show you a little bit of GEO. So, if I'm looking for data, you can often go to a paper, and a paper will have a GEO ID attached to it. Probably find a paper. See how right I am. Is there a GEO ID in this? Nope. I'll find one of them. So this is GEO. So you can have free text GEO. So I'm going to go MIC CH12. So I'm looking for the MIC transcription factor, CH12 cell line, ChIP-seq. 24 results. Open them up. And here we can see we have a nice search page. We have some direct samples there, and we have whole studies being described here. So I'm going to go to this one. You can see it describes the experiment, the organism, when it was submitted. It has, for the whole data, a download here, but this doesn't contain raw data. This contains the process data. So it's got the BAM file. We just learned about BAM files. It's got bed file, bigwig file, a broad peak, and narrow peak. I've got no idea what the pair is. And then this will be for the overall experiment. But if I wanted to go find what I was interested in, so CH12, I was interested in MIC. There it is. Going here. It tells me a little bit more, and this type of stuff you get only in GEO. Characteristics of the actual cell line, the treatments which were applied. We often get some description of the extraction protocol, instrument used, data processing, but this is all free text. It is not controlled. So if I one day decide I type in a very loose description of my processing, I will be able to submit it and it will be there. It's no one's enforcing this. So it has more information, but it's not really controlled what people put. This looks good because this is encode. So now I get in here, I can get some information per sample. If we can't quite read this, this is our narrow peak. So this is the actual positions of where this transcription factor is bound. And this is the bigwig. So this is the actual signal over the whole genome. How much signal do we have for this particular ChIP-seq mark? How much binding do we have? GEO is cool. It's often where you'll submit your data to, fill out some forms, but it's not particularly regularized, standardized. So the short read archive is NCBI's high throughput sequencing specific repository. And it is built for sequence data. Slight problem with SRA is that they decided to create their own format for fastq. So this is problematic because fastq won the competition. It got adopted by all these aligners. So for a long period of time, effectively, you had to use SRA's own toolkit to download an SRA file and turn it into a fastq, and then you could start processing. So that was actually one of my biggest problems with SRA. No longer true. They've actually fixed this up quite nicely. Oh, I guess SRA. Sorry. Let's do this. Yeah, I can do CH12. But we find the same experiment here somewhere. And then if it's got a little bit more, no thanks, no survey, please. It's got some of the description taken straight from GEO. Importantly, if I click on the run, there is no data here which is processed. It's only raw data. No thanks. You can already see something here. We'll come back to this in more detail. Those quality scores I showed you, those Fred quality scores, they're actually displayed here, like an overall graph of it. You can see, oh no, it says Fred quality score. You see most of the read quality is up here, but there are some reads which are bad. Right? So we would use those Fred quality scores to cut out maybe this bottom set. Over here, they're showing the whole distribution of the quality of their data. So this gives me some information. Most importantly, somewhere here, I can go Fasta and fastq download. That didn't used to be available. I can click there and I can download it as a fastq. I can download the reads as a Fasta. Why I would want to do that, I'm not sure. But I can just click that and it will go and download these as a Fasta. So that's SRA, which is a lot like, sorry, actually really lost my thing. So that was SRA, which, whereas GEO is the US general repository for both microarrays, expression data, and high throughput sequencing, GEO actually takes advantage of SRA. So when you submit to GEO, it actually SRA, which is holding your raw data. And we can just download it directly from there. ENA is the British equivalent, European equivalent, not British anymore. We're out of Europe. Browser. And again, it's very much like SRA. It allows you to do a free text search, CH12, Nick. It's a little slow. It is going to pull me up some results, hopefully. This happens. Study project, study. And then it will have lots of information here. It's a little slow. But what's nice is it gives you this kind of table format. So I can look up a whole study. I can even take my SRA IDs and just search them in here. What's really useful for me is that if I want to, I can click this button TSV, and all the information in this table will be downloaded to my computer, including the full path to these fastqs. So just to work programmatically, rather than having to click and download the fastq to my computer, if I download this TSV, it's going to give me all these paths, and I can actually just use R again, read in the file, loop through those rows for that column, and download the files one at a time. So in reality, it's kind of GEO will contain the most text information about the experiment. SRA is just going to contain the fastq, and ENA compromises a little bit by showing information on both. I would say provides a slightly nicer table to retrieve the data from. So apart from these three major repositories, we also have sites which are built around particular projects. And often these will have a lot more information and a lot more processed data than things which are more generic. GEO needs to handle everything, so it doesn't necessarily force us to have all these different processed data types. In fact, when you submit to GEO, they just ask for any type of processed data. They don't care what it is. They just want something. ENCODE, which hopefully people know, is a fantastic repository of data from the ENCODE experiment. Ooh, it's updated quite a lot since I was here. But you can often just search your experiment here. Let me try and find one. Jeez. Okay. We get to these pages in ENCODE, and these are fantastic because they don't only contain like information and links to their actual downloadable files, but they also provide links to multiple different types of process files, like bigwigs, bed graphs, peaks under different thresholds. And they even give you a little linear genome browser to review your data in. Yeah, but here they've also got the full documentation for how this data was processed, which is very cool. Not necessarily super easy to follow along, if I remember right. Nope, no. A very simple pipeline overview for that particular one there, I think. But yeah, so here we go, file details. And in here, you can see you can download all these different file types for this particular experiment. And what's really nice about ENCODE is every single experiment they run has the same file types processed and generated. So it's very useful. Apart from that, I think we have just a few more things to talk about. So the ENCODE portal. Yeah, just to say, there are other specialist repositories that exist. One which is really useful and worth mentioning is Recount 3. It's no longer Recount 2. What Recount 3 does is similar to ENCODE, but they've gone through most of the experiments which is submitted. They run through it and they produce gene and exon counts for you. So you can just download a table, an Excel file of all the gene to sample counts. And that's what would go into a standard RNA-seq analysis. So you're skipping out the step of processing, aligning and all that. They do it in one standardized way for you. But you also have very specific things like ImGen, if you like immunology, body map, if you're trying to find a specific expression, and expression atlas as well. I want to know that this is expressed only in this tissue. I can go to expression. Final thing we didn't cover here is where do we get our reference genomes from? Okay, and there are lots of different places you can do reference genomes. Like I said, we have major revisions, minor revisions. We have to get our genome sequences faster. Gene builds as GFF and GTF. And one of the problems people face is that they download their Fasta from UCSC or Ensemble. They get their GTF off their friend because they're telling them that's the one we use in the lab. And then you end up with mixed sources of annotation. This can be a real problem. Often you'll lose track of where I got this GTF. iGenomes is by Illumina, and I think it has a really nice solution for this. These files are big. They're like 60 gigabytes to download. Okay? But what they have is for every major genome build, you can see here, you can either, if you click this, it will download a 40 gigabyte zip file containing the Fasta, the GTF, the genome annotations, the rRNA tracks, any annotation you need for your genome. They have deliberately put it into one package, and they make life a lot easier for us to have to download multiple separate files. And so I recommend if you're going to do some of your own analysis, it takes a long time to download, but you can just click that, and that will download it to your computer. I'm also going to show you how you can extract these programmatically in ARM as well. So we're going to get package and version controller. So that's the end of our first session today. I'm going to stop my share.